Welcome to ISL-enabled Virtual Lab of CSIR.
Hi, I'm Saikat Lab, a senior scientist at CSIR CCMB. So the research that we perform in our lab actually looks into how different macromolecular assemblies assemble inside the cell, and then they perform different functions. More specifically, what we are interested in looking at is how different cytoskeletal elements, which provide support structures for cell, as well as they provide the necessary protrusive forces needed for different functions inside the cell. So we look at how these actin cytoskeleton assemblies are assembled inside the cell in response to different signals. More specifically, what we look at are some of these nucleators that initiate the process of actin assembly. So in our lab, we are mostly interested in looking at one of the complexes called the ARP23 complex, or the ARP23 complex, which is one of the primary nucleator that helps in essentially forming a dense branch network of actin inside the cells. And that is important because that way cells actually distribute forces towards the membrane or wherever it requires to do different functions. Now, why is it important to understand these processes? It's critical because they are the underlying force that is necessary for cells for doing their function. But also these are harnessed by various pathogenic bacteria or viruses or for their onset of infection, or any of these deregulations are primary hallmarks of cancers or any developmental disorders or neurodegeneration, for example. So what our goal is to look at a very high resolution using cutting edge technology like electron microscopy at the different molecular interactions that finally manifest itself into these nucleation processes, or how these different architectures of actin are essentially formed inside the cell, whereby we can then develop therapeutic molecules or drugs in future, which will help sort of cure some of these diseases and stuff. So in short, what we try to understand is how these Lego pieces or these little pieces are assembled in the cells in response to signal by using some of these cutting edge technology like structural biology and biophysical techniques, as well as cell biology, for a better understanding of how these molecules work inside the cell, so that we can come up with therapeutic molecules and cure for diseases in future.